Okay, last lecture of the unit. I'm doing them all in a row. So 4, 5, 6, all on the same day. Sorry if it's, I don't know. I'm tired. I need more caffeine. Phylogeny in the tree of life. Phylogeny is, well, we'll talk about it right here. A phylogeny is showing the evolutionary history of species or groups of related species. If you ever had to learn all the like orderings and classification patterns of animals, it's like kingdom, phylum, class, order, family, genus, species, I think. Phylum is related to phylogeny. So it's just grouping things. Taxonomy is very much related. It has to do more with naming and dividing them up. Phylogenies are generally used to show like a group of related species and traits or something, whereas a taxonomy is more organization tool, but I'll probably use a little bit of both interchangeably. They're essentially the same thing for our purposes. Talking about how a phylogenetic tree is set up, go laser pointer mode. So taxons are individual branches. They represent a single species. The closer two taxons are, the more closely related they are. If they're right next to each other, we call them sister taxa. And then you have this one at the bottom, which can either be called the basal taxon, it's like the base taxon, or the outgroup. Either one will be used. Anytime you see a branch, that usually means that's the last common ancestor that these things shared. So we can say taxon D, E, and F shared a common ancestor more distantly in the past than taxons B and C do. At the very left, you have the initial branch point that is the most recent common ancestor of everything here. Horizontally, generally represents time or an accumulation of genetic differences, which is why we can say D, E, and F split off more distantly in the past than B and C did, so I could make the argument that B and C are more closely related than D and E, for example. How do we even make these things, though? It does come back to morphology, which is the study of shape, how things are shaped. So we use morphologies, looking at the structures, looking at fossils. More recently, we have been able to incorporate genetic data as well as biochemical data, like what proteins are they making, what nutrients do they need, things like that. As a reminder, homologies are similarities due to shared ancestry. The more similar it is, the more likely you are to be closely related, where you'll have analogous structures, which are similarities that are due to convergent evolution. Remember, convergent evolution is just similar environments will produce similar adaptations. Sometimes fins are just good for swimming. Even if you're not closely related, you may all evolve very similar shaped fins just because that is what's good to move through the water. But you have to be careful to not confuse analogy with homology. That is generally where DNA sequencing is going to come in. I think that's a video that we'll watch in class. Why do things keep evolving into crabs? Spoiler, it's because the crab shape is just a good shape. We'll talk about that more in class. A few more terms before we take a look at an example of what we use to make phylogenetic trees. Sometimes you'll see this as the study of cladistics, which is grouping organisms based on their common descent. So we have taxonomy, phylogeny, and cladistics, all of which are kind of the same thing. Not really, but they're all just ancestral trees, essentially. You will see the word clade. This is the group of species and all, including the ancestor and all of the descendants. So what we looked at a couple slides back would have been a cladogram, which is just a thing showing a clade. And then what we're looking for to make a cladogram are shared ancestral characters and shared derived characters. So shared ancestral characters are traits that exist in all of them, from the common ancestor all the way through each taxon. they're characters that every single thing has because it did originate in the ancestor and it persisted. And then you have derived characters, shared derived characters, and these are new features that are unique to specific branches or subclades. So if a particular subspecies or other species in a group evolves wings, then that would be something that separates them. So you're kind of looking for when did new traits emerge. If there is a new trait that emerges, we generally consider that to be when a new species will split off, and anything that has that new trait likely split off around the same time. And then finally, we have the outgroup, the basal taxon. It's the least closely related. But let's look at an example here. So on the left is a character table, and on the right is a phylogenetic tree, cladogram, whatever you want to call it. This one is comparing lancelets, which is a little flatworm, to lampreys, to bass, to frog, to turtle, to leopard. So going from a pretty basic thing all the way up to mammals. And the character table is basically organized so that the outgroup does not have any of the traits that you're looking at. And you slowly add in traits one by one. So the lamprey is the first thing. Everything else has it too, but the lamprey is the first thing to have a backbone. It has the backbone, it doesn't have any of the other characters, any of the other traits. So that's why we put the lamprey splitting off first, because it only has this one character here. We look at the bass, it has two. It also has a hinged jaw. So we will say that the bass was the next thing to split off. We keep going. Frogs have 3 of them, so they're next to split off. Turtles have 4. Shout out Momoni. So they will be next to split off. Amnion, by the way, is it's like a protection for the egg, essentially. It's like a membrane around the egg. And then lastly, a leopard is the first thing that shows hair, fur, and so it has all the traits, and so it must be the most recent thing. As evidenced by, it has the shortest line, it's the most recent branching off point, and so at the end, we just have a series of little branching offs. The lancelet had nothing, it branches off first, and so its line is the longest, meaning hasn't changed in a while. It's pretty closely related still to that common ancestor that gave rise to these other things. The lamprey gets one new trait, splits off, and we just get newer and newer traits as we go till we get to the leopard, which has the most of the traits, meaning it is the most recent to evolve. We will practice this in class. It is a pretty major skill for the exam. Let's talk about branch lengths for a bit. Often, the tree that's drawn will have proportional branch lengths. Sometimes they'll actually give you a timeline down below. They may not always. But if they're ever asking you questions about like how long ago things split off, or like for this one, it could ask, what is more, has a more recent common ancestor, a human and a chicken or a human and a frog? And you just go back, human and chicken is less distant than the frog. So the longer the branch, the more time has passed, the more genetic changes that have happened, because we consider mutations to kind of happen at a constant rate. So longer branch, longer time, essentially. But ultimately, these trees are hypotheses. They are our best guesses that fit the most data based on morphological, molecular, or fossil data. But they are constantly being updated, constantly being redrawn. There's not even a right way to do it with the data that we have. It depends on how important you consider each factor, which is somewhat arbitrary. But what it does let us do is it lets us predict features of an ancestor based on the features of its descendants. So for example, we can look at the descendants of dinosaurs, and if we look at them and they all build nests and brood and like sit over the nest, watch the eggs, if all of the ancestors do that, then we can infer that the dinosaurs probably did as well. We can't know 100%, nothing in biology is ever 100%, but we know likely that dinosaurs would have had these behaviors too, because their ancestors have them, and it didn't emerge in the dinosaurs. So, it's a hypothesis, it's a good guess based off data, but they are not absolute. They are, however, based off of this idea of evolutionary time, where essentially we kind of assume that the rate of mutation and thus evolution is kind of constant over time. That if you give an organism 100 million years, you should expect this many genetic changes. We can plot this. It's a pretty decent line of best fit. And what it shows us is, again, the rate of evolution of new species of mutations seems to be pretty constant. This lets us do some cool things and cool predictions. For example, with HIV, we were able to apply this molecular clock. We know HIV is descended from a virus that could infect other primates. But the data that we have shows us that HIV likely spread to humans more than once. Historically, it showed up in, I think, the late 70s, early 80s is when it started the AIDS crisis. But if we use the molecular clock analysis, it shows us that HIV likely spread to humans in the 1930s, like 30 plus years before we traditionally think of it. And there is some error with this. There's like the darker the line here is more likely. As you go out, it's less likely that it came in those years, but it probably showed up before we expected it, which is just kind of an interesting fact. The last thing I want to talk about is kind of on this idea of all of this is temporary and probably only 80% right, because we're working with bad data. We don't have, sorry for the stretch, all of the organisms, we don't have all the fossils, we are never going to have all the data that we need to actually do this task, which may not even be possible if we did have it. In the 10 years since I was in high school, we've invented a whole new classification system. When I learned it, the highest level was kingdoms, which was like plants, animals, fungi. Then we actually had things called Monera and Protista, neither of which are really used anymore. And now we've gone up even further to this domain feature where you have eukarya, which is all of the eukaryotic cells and organisms. You have bacteria, and then you have archaea, which are archaea bacteria, all coming back to a common ancestor. What this implied that we were able to see, which is kind of cool, is that we are more closely related to archaea than we are to bacteria. So we were able to, with this new grouping, kind of infer more of how complex life emerged. However, in 5 years, this organization system probably will be changed because we always find new fossils, new data, genetic sequencing becomes better, and we're able to make better conclusions. But the tree of life is ultimately messy. You can probably connect. The tree of life is messy. You can never do it the right way. You're just trying to fit things into categories that do not necessarily belong there. But that is it. That is the last lecture. And that we'll probably do lectures in ecology. Sorry. Maybe 1 or 2. Not a ton. We're just going to spend a week on it. That's all. Test. Spring break soon. You can do it. I believe in you. Rah, rah.